Okay, so I think a kind of basic question for anyone reading Title, like why would you use DNA to build molecular machines? Just put why DNA. So at the scales that we're trying to build things, so typically like with 10 to 1,000 nanometers, I don't think there is any alternative that has the same precision that we can engineer systems with that kind of precision. And then I would say that DNA provides control. So we have very fine control via complementary base pairing. So we can specifically interact with different part of the machinery, and we can do it reversibly via low-volt mediated stranded baseband. And then finally, like I think one issue with DNA is it's not a very functional material, it's kind of boring material, but that's not natural DNA. And in synthetic DNA, you can incorporate like a lot of other base pairs. So that's very commercially available. You just go to a DNA synthesis company, you have just long lists of things that can replace their own base pair with to make the material more interesting. And that sort of includes sort of attachment chemistry and linkers, so you can do further processing and add other stuff afterwards to your DNA. Okay, so how do we make it? I put in this slide very quickly. So we use a technique called DNA origami to fold DNA strands. So the idea is we decide on a shape that we want, and then we rationally design sequences of short DNA strands, and then we just order them from a DNA synthesis company. We mix them with a long natural DNA strand, and we anneal it, and then hopefully it assembles the shape we want. And what's really important to mention here is that we get control over where every staple strand ends up in the structure. So this structure might be composed of 200 oligonucleotides, but we know the position of each one of them. And if we add something to them, we have the chemistry stuff, then we know where that piece of chemistry ends up in the final structure. So we can control the position of other molecules with very high precision on top of the structure. Okay, no, I think I skipped this slide. So I'm just saying there's been tons of different DNA origami mechanisms over the last decade. So there's been these kind of simple sort of switches and joints and rotary motors and sort of cages that can open and close. And these things have been able to actuate via a bunch of different ways. For example, introduction of DNA strands, electric fields, magnetic fields, and also sort of binding to interesting targets, biological targets. So this is a relatively big field, I would say. And the goal of our project is just seen in this kind of simple sketch. We want to create a very simple printer. So like the simplest possible 2D printer you can build. So in order to do that, we need to make a 2D positioning device where we can move a right head in 2 dimensions over a surface. And then we need the right head to somehow be able to pattern the surface. So how we started with it was just like the most simple type of DNA motor you could build, which is a linear actuator. So we had something that looks like a rotaxane, I guess, in molecular machines chemistry, but bigger. So it's around 200 nanometers long. And it's composed of 2 DNA origami components. So this rotary in blue linked along a beam, a rotaxane along a beam where it's topologically locked. And then it's free to diffuse along the beam. But if we add specific address strands, we can drive it into one position in 1 dimension. And oh wait, no, this will end up in So the question is like, how do we take these very simple 1D motors and make it into 2D device? So we followed 2 approaches to take 1D into 2D, and it's kind of inspired by macroscopic scale. And I think that what we call the Cartesian positioning device is the most similar to what you might see in a macroscopic device. So you have, again, the rotaxane linked on a central beam where it can move in 1 dimension, and then that beam is locked in 1 dimension on the base beams. So it can go like this and this. That means that we need 3 linear motors for this to work. And then a slightly simpler approach is a polar positioning device that looks like a bow and arrow. And here we instead get away with using 2 linear actuators to get 2D motion. But I'm mostly going to talk about the Cartesian positioning device.
Yes, of course.
So this is kind of the DNA origami application, what they would look like by some type of simulation, how we imagine they would look like, in our DNA designs. So typically you're kind of limited in the size you can do in 1 DNA origami structure. So somewhere 5 megadalton or like 100 by 100 nanometers, something like this. So when you go bigger, you need more structures. So in this case, we needed to build 3 separate components that we link together, or 2 separate for the polar positioning device. So what we did is we just assign them separately. We assemble them separately, you have to purify them, and then we mix them and purify the mixed assembly. And then we could see them. You can see the final assembly in electron microscopy and also in DNA paint, so the super resolution microscopy technique. And that's going to be quite a lot of DNA paint data here. So the resolution is much worse than electron microscopy. The nice thing is we don't have to destroy the structures. We can work in a flow cell, we can feed stuff in, we can image them again, which is very nice about sort of light microscopy. So this is just kind of a slide showing kind of the design consideration you have to have. So figuring out how to link. So we have a central bit, the canvas here, and these 2 base beams, and they need to assemble via these linkages. So the canvas keeps these 2 beams parallel with each other, and then they're also linked along the bottom beam. And this is quite difficult to design if you don't have simulation. So we have Michael, who was sitting there before, who is very good at simulation. We'll talk more about it if you want to simulate DNA. Anyway, this is kind of how we ended up imaging the final device. We can with DNA paint, we choose what we image. We just image different DNA strands, and because we can program where the DNA strands come out of structure, we can program what we image. So in this case, it's changed to having DNA paint strands along these 2 beams, and then along a line on the canvas. That's to get us kind of a nice coordinate system. And then we also have orthogonal DNA paint strands that we can image separately on the slider. And the way we make it move is we have these unique algebra strands that protrude from these base beams, and also this beam going here. And they are designed not to be orthogonal with each other. So we can feed in specific binder oligos to force the sleeve to bind in position on the rail, and then separate binders again to force the rail to bind on these kind of base beams. And that's how we get 2D positioning in this system. Oh yes, this is kind of just like practical concerns, like what is it like working with DNA? So it doesn't always work. And initially we tried to just position it. So we tried to position the slider in the middle of the rail here. And we saw that the rail arm got pretty well positioned, but obviously the sleeve didn't work very well. So this is like an average image of hundreds of devices in DNA paint. We can see that actually it kind of got stuck on the loops. So this is the issue. We have the sleeve topologically locked around this base beam, around this rail, but unfortunately can thread off to these loop regions. And if we clamp down the beam, then the slider is stuck in the top and bottom. But fortunately, we realized if we sort of just pump up the magnesium concentration, we're more likely to drive the sleeve back on the rail. So just looking like, can we position it in 2 dimensions? So we take hundreds of images of the structure and average them to get a feeling of what the precision is. And it seems like, yes, we can position it in 2D, but the precision is a bit of an open question, I would say. And then it's kind of a question, can we reposition them? Can we release the linkages and then relink it somewhere else? I would say, yes, we can do it, but we need to get better at it. But yeah, we can move it in 2D. So actually, the precision of the device is kind of difficult to answer from these images because the microscopy is not that high resolution. So we can try to use simulation to get a feeling of how much is the sleeve moving on top of the canvas. So this is just a nanometer scale. So you have a motion along this axis that's on the order of 10 nanometers, and you have around 5 nanometers of motion in this axis like this. So I think it's pretty obvious that we need to improve the fit of this device. So get tighter fits of all the components, and we can get less flexibility in the system. Yeah, so finally what we do is we want to see, can we turn this slider into a right head? So we need to make the slider somehow functionally interact with the canvas down below. And we chose to do it from an all DNA system because DNA is basically what we work with. So we worked with this kind of tow hole catalyst system. So we placed just a special DNA strand below the slider, that's capable of interacting with an array of pixels, acceptor pixels that we put on the canvas. And then as we move it in, since we're working a flow cell, we can move it into position over a pixel and feed in ink via flow cell, so that gets preferentially incorporated in the pixels that's below the right head. So you would put essentially like a pixel array, 56 pixels on top of the canvas to get us something like 60 by 72 nanometers to work with. This is important. And then this is essentially it. So we put this is how we imagine it working. We have this array of pixels. We have a right head DNA strand below the right sleeve that interacts with the pixels to get them to interact. And then just looking at what does printing look like. So we can lock the slider in a corner, for example, and then just sort of flow in more and more ink to see how much ink do we need to achieve patterning. And it seems like when we put more, we get more patterning, but we also sort of get more off-target effects. So we can do patterning, and we can also sort of move the slider. We can do patterning in one position, move it to another position, and then do patterning again. We can see that we sort of printed these very, very simple patterns on top of the canvas. But that's how far we got. So this is kind of the interesting part. Like, what do we need to get better? So we have kind of design lessons. We need to get more rigid, and I think we still do that within DNA if we just improve sort of the precision and fit of our components. And I think we want to do something more interesting and write DNA on DNA. So that's something that we need to think about. And then we also want to think about what are we trying to print on? Should we try to print on DNA? It's probably not that useful. So it would be nice if we can remove the canvas after we deposit it on a surface to work directly on glass or metal or whatever we want to print on. So this is very open, and I just put together these slides coming over, and I have thought less than you might think about what we should try to make with it. I think like these are the sort of the main design constraints. So the drawbacks is we sort of have to be working with DNA, so we probably have to work with chemistry that can work in mild conditions. So something like click chemistry, DNA hybridization that we're doing now, or enzymatic reactions. That should work fine in our buffers. And also, at least now, our workspace is pretty small. So that's our kind of constraints. But I think the benefit is it's massively parallel. So we can have billions of devices stuck on the surface but they have to do well, they don't have to do exactly the same thing. We could also program that. And I think we could also work on surfaces that are not perfectly flat compared to lithography, where you need very flat surfaces. And then there's kind of the open question: what should we try to pattern? Should we retain the canvas as a kind of work surface, or we should get rid of it, try to pattern directly on a surface? So I think these are the very, very rough ideas of what can be made. So I think either you try to retain the canvas and use it as a 2D molecular printer or like a 2D ribosome. So you would have some type of chemical element stuck in the middle. So that's your sort of starting ground. And then you move around. You attach the next block to the right head, or you have a catalyst on the right head. And then you put it into position next to where you want to link it into the molecule. And then you perform a chemical step so that these two get hybridized. You release the right head from the first building block, and then you can recharge the right head with new ink. You can sort of move wherever you want to print in the new block. And I think sort of I'm not the chemist, so the easiest thing for me, of course, is to work with DNA blocks, but it's not that interesting. You can also imagine working with sort of larger peptide bits or like polymer bits. So we don't have a very high precision yet, so we're not going to do like single atomic manufacturing, but I think we can do interesting stuff. And then if you remove the canvas, you can imagine doing something like 2D meta surfaces. You just like put in gold nanoparticles, something wherever you want. And then you could imagine working on a DNA brush. You could have a glass slide with a DNA brush on it. You put on the printer and you print the pattern you want. You get like a massively parallel copies of the same pattern. And of course, you can imagine sort of linking the printers to each other if you also want controlled arrays of your particles, patterns on the printer, on the surface. And finally, I just want to thank everyone we work with, especially Rafa and John, who did a lot of the work, and our collaborators and our funders. Thank you.
Yes, go for it.
Hey, this is a super cool technology. I noticed you're in Oxford. I'm there as well. We could get copies sometime.
Okay, yeah.
I'm curious,
Yeah.
the Department of Energy is funding you. What's their interest in this kind of technology?
So they had a big research call around 5 years ago in atomically precise manufacturing. So it's a specified call, and that's how the funding came in.
But they didn't say what they intend to use it for. We're just curious about it.
No, I think so. Actually, Eric Drexler had some sort of connection with the Department of Energy. They had some sort of brainstorming session, and then this sort of one-time call came out. And I think it's, I mean, in that case, the idea is to drive innovation in the US, I guess, because we were just a subsidiary of a group in the US, so we got sort of co-funded through that.
Cool. And have you guys thought much about spinning out this technology into a company, into a startup?
Not too much. We published the stuff without patenting it, so we didn't think too much about that. And I think I'm thinking more about it now than I did then when we published it. But yeah, I think you need to make something useful, and I don't know what that is yet. So I think it's yeah.
Because I mean, it's nice to open source stuff, but I'm in the pharma space, and if we were to open source stuff, nobody would spend any money running the clinical trial. So what you can do is you can spin out a company and raise tens of millions of dollars and funnel that back into your lab, rather than relying on the government research agencies that are a little bit more miserly with the money. So yeah, I'm there in Oxford. I can give you some tips on how to spin something up. Oxford's a difficult university to work with, though, because it's a real pain in the neck for technology licensing, but it can be done.
So I have moved to Stockholm as this year. I'm still affiliated, but now I live in Stockholm working on slightly different stuff. So
cool. Well, that's even better.
Does it make your job easier?
It definitely does, because in Sweden there's something called professor's privilege. They're trying to get rid of it, but basically Sweden is one of the only countries where it's still this way, where the professors or the inventors own the IP completely, rather than the university owning the IP. And that may be one reason why Sweden has such a high incidence of startups, even though it's not a very startup-y sort of culture, because of professor's privilege. So that definitely makes things easier.
Yeah, for sure. Yeah, I think I'm thinking more about patenting stuff now than I did then.
When we build molecules, it's quite straightforward to say, I want this part to be rigid, this part to be more flexible, like thinking not in change, I want to make it flexible, a more like aromatic part, if I want to make it rigid. But you work with like DNA, so you're kind of limited to what building blocks. How do you actually, because I see that you mentioned about parts that are rigid, parts that are more flexible. Like, how would you like compare these two systems? What do you use to make these two properties?
I mean, that's a huge issue working with DNA. It's relatively flexible. I mean, but if you build these kind of beams, like a DNA origami beam, that would have a persistence length of like 10 micrometers. So actually, the beam itself is flexing quite little at the scales we're working at. But then we have these DNA linkages, and that's causing a lot of issues. So when you actually simulate it, you can try to do design lessons, try to link it in harder, but yeah.
There is a solution which comes from biochemistry, which is DNA crosslinks. So there are enzymatic and non-enzymatic, like covalent reactions that will make it more sturdy, less flexible. And this actually happens and drives the aging process within us. Like UV radiation or reactive oxygen species damages the DNA and crosslink.
Yeah, actually in DNA, there's been work in DNA to do what they call welding, essentially. So they put these thymines at the right position, and then they hit it with UV and link it. And then there's also work in sort of biomineralization, so you can just coat it in a layer of silica. But the problem is you get rid of a lot of functionality. I would say in both approaches of functional, we need to be able to address the strands to drive the positioning. And if we coat it or cross-link it, we might get issues with that. So it's like a kind of difficult trade-off. Yeah.
Super cool stuff. You know, this is I joke, have you guys heard of the term food porn? You know, like there's a photograph or video of food, you know, right away. You know, it's like you watch the food channel or whatever. Well, this is futurism porn. And for me, you know, like this kind of precise molecular manufacturing is the most futuristic stuff that I can think of. Super cool.
Yeah. I mean, it's very, and that was so. I think the grant call was atomically precise manufacturing, but we're nowhere near that. But I think you just have to start somewhere. I mean, And then maybe it's not going to be DNA that makes things better, but you just have to start somewhere, and then you will learn lessons. And I think you can probably make useful things even working with bigger building blocks than individual atoms.
May I ask, what is the future of this project for you, or how can we all help?
Yeah, so I'm actually working on other stuff right now, which I quite like. So I haven't really worked on this for a year. I'm working on more bio stuff now and sort of sequencing things, and I find that to be pretty fun. But I don't really have any. I don't have any funding to do more manufacturing, and
So that would maybe help.
yeah, I think so. I think when I was kind of done with it, I didn't want to see it again.
Yeah.
But now, sort of coming around, like a while later, I'm getting happier about it, more enthusiastic again.
Yeah.
So,
So we should give it its time.
for sure.
Okay. Yeah.
Yeah.
Perfect. Did we have any other questions?
You're a sequencing guy. What kind of sequencing stuff are you working on?
Can't say.
No, I'm working on sort of spatial transcriptomics techniques, and also, also evolutionary DNA nanostructures. The idea to have a sort of large library structure we can read out the shape by sequencing, so to sort of move away from rational design to evolutionary design.
What do you think of that?
We're doing something different. So we're trying to predict the sequence from images. You just take a picture of whatever tissue or cell or you. How can we predict this information?
Wow. So wait, you take a picture of what exactly?
A cell, a tissue, or whatever living organism. How do you actually predict the genomics information from those pictures?
Wow.
Which I will talk later.
Thank you so much.
Thank you.